In case y'all are still wondering why I'm so behind in grading, it's because I can barely get lectures done without getting interrupted. Um, so I did not mean for there to be a break between the last one and this one, but I paused my recording and then my connection to Zoom was lost. So I was in mid-sentence, I believe. So we were talking about, I was just saying that cystic fibrosis is a recessive condition. And I was talking about um, the pattern that many genetic disorders are more prevalent in certain ethnic groups. And so in the case of cystic fibrosis, it's more common in people of European descent, right? So white people. Okay. It's more common. Yeah. So if you, depending on what ethnic group you are a part of, you get asked different questions if you are going to have a baby, right? To decide if you need to have any genetic testing, right? So, as a white lady, I got asked if I have any family history of cystic fibrosis, and a couple of other different conditions that are more common in my particular background. If you are a person of color, or if you are of Jewish background, right? There's other disorders, right, that are common in those groups. So anyway, okay, so I'm just putting that out there so you kind of know. Okay. All right. This is not to say that people of other ethnic groups can't get these, right? It's just they're more common in certain groups. But of course, now that we're doing so much moving around and we meet people from all over, right? Increasingly these things are going to become more, widespread in different groups of people. Another example of a disorder that is something that is, could be viewed as harmful, is one type of dwarfism called achondroplasia, which is a dominant condition. There are lots of different types of dwarfism, first of all. Okay, there are lots of different causes for dwarfism. This is just one of the causes of dwarfism. This particular type of dwarfism causes the limb bones, arms and legs, to be shorter than normal. But like the trunk of the body and the head are generally average sized. There are a few different actors that have this particular type of dwarfism. And one of them, I can't think of his name right now. He's in that show that everybody likes. But I'm all distracted because of my kids, so I can't think of his name right now, which is irritating. I'm gonna think of it tonight when I'm trying to go to sleep. Anyway, okay. This particular condition is also not common, right? It is a mutant trait, but it's a dominant trait, right? So people who have this particular type of dwarfism are heterozygous generally for this condition. The vast majority of us that are of average height are I don't like normal, are average height, are homozygous recessive for that particular gene. Okay. Some other characteristics. Part of the reason why we're talking about these is because of the lab that you're going to do next week. So because genetics is complicated, I like to spread it out a little bit. Okay. And so so some of this stuff you're going to want to refer back to for lab. So there's some images of, just more traits that humans have. Okay. So freckles, no freckles. A widow's peak. A widow's peak is when you have a little point. So I don't have one. My problem with my hairline is you look at my hairline and it looks like there's a little bit of an indentation right there, but it's a straight hairline. It's just not an even hairline, especially because now that I'm going gray, it looks extra white right here. So a widow's peak is when somebody has a hairline that has a somewhat distinct point as opposed to just a straight hairline. Okay. Widow's peaks happen to be dominant. Not better or worse, but they happen to be dominant. Okay. And then the whole free versus attached earlobe is another characteristic. Now, these are the characteristics that we're focusing on because these are really easy to see. And so we're going to use these in our lab. So a free earlobe, right? See how her earlobe like tucks next to the side of her head like that? So I have free earlobes. Let me move my speaker or my headphone wire, right? So see how it comes around and then it tucks up like that? That's a free earlobe, okay? An attached earlobe is when your earlobe just kind of merges like that. So it doesn't have a little like tuck. You don't have a flappy earlobe, okay? Once again, one isn't necessarily better or worse than the other, right? But free lobes are dominant to attached lobes, okay? Another fun one that you're going to look at in lab is whether or not you can roll your tongue. Okay, so this is another one that the majority of people can do this. People who can't do this, it's kind of adorable when people try to do it and they can't, because it doesn't matter what they do. They just literally just can't do it because they don't have the muscles necessary to do it. So whether or not you can roll your tongue. Okay, so see how I'm making a taco with my tongue? That's tongue rolling. People who can't roll their tongues, this is what they do when they try. They go. It's adorable. Okay. So once again, dominant is being able to roll your tongue. Recessive, no tongue rolling. Okay. Having a cleft chin, right? So do you have like a dimple in the middle of your chin? Okay. Yes or no? Right. This is another earlobe situation. Here's another example of a widow's peak versus straight hairline, right? So Leo DiCaprio, right, has a widow's peak. Okay. All right. One other, since we're talking about humans, okay, one other thing, terminology, one other, vocabulary word that we're introducing now is the concept of a carrier. So we use the term carrier. Generally, what carrier means, so carrier usually equals somebody who is heterozygous when we're talking about a recessive condition. Okay, so whatever A stands for, let's say not achondroplasia, because that is not recessive, that's dominant. Let's say it's albinism, even though really we should be using N for normal pigment as the dominant. Okay, but let's say that A, and I'm just totally making this up, right? Let's say A is normal pigment, big A is normal pigment, little a is albinism. Okay, if somebody is homozygous dominant, right, that is the normal condition. If somebody is heterozygous, their phenotype is going to be normal, right? They're going to have normal pigmentation. That's what their phenotype is going to be, right? But we might refer to them as a carrier because somebody who is a carrier can have offspring who are homozygous recessive, which would express whatever particular trait or condition that we're talking about, right? So we have a normal appearance carrier male, a normal appearance carrier female, even though neither one of them are expressing the albinism trait, right? There's roughly a 25% chance, because they are both carriers, they are both heterozygous. There is a 25% chance that they can have a child with albinism. Okay. So when we talk about a carrier, where your brain should go when you hear the word carrier is, oh, that's probably heterozygous, right? But essentially, what it means is somebody has the alleles for a trait, but they don't express that trait. Okay. All right. Okay. So the next thing here on our little to-do list is to talk about pedigrees for a hot minute. Pedigrees are interesting. So why do we do pedigrees? What's the symbolism that's involved in pedigrees? All of that. the first thing you need to know about pedigrees is these are especially useful for people. You can do them for other organisms. So, right, if you're somebody that's really into like dog breeding, for example, one of my friends has gotten really into this in the last 10 years. Um, and so she has like a detailed pedigree of all of her dogs, like who their parents were and who all their siblings are. And she can like trace her dogs to, anyway. Okay. She's way into it. So you can certainly use them for other animals as well, racehorses, dogs, stuff like that. Okay. These are especially useful for people though. And the reason that they're useful for people is before genetic testing was a thing, right? Let's say you were trying to figure out, well, what is somebody's risk of, having a child with cystic fibrosis? Well, nowadays they do a genetic test, right? They're like, hey, do you think, do you fit the, group that might be at risk for this? Okay, we're just going to test you, right? Okay. But back in the day, right? How did you figure out if somebody was a carrier for a particular condition? Well, with our dog example, we went ahead and just did a test cross, right? So remember the test cross where it's like, I know that you have the dominant phenotype, right? I know that she has the dominant phenotype. The dog here is black, but I don't know if she's homozygous dominant or if she's heterozygous. I don't know. Someone just made her with another dog. You can't do that with people, right? So you can't say, hey, Sally, I think I'm really curious. I want to know if you have, if you're a carrier for a particular condition. So why don't we just go ahead and pair you up with this guy that I know is homozygous recessive, and you guys can have a bunch of babies together, and we'll just see. We'll see what happens. Okay, that's preposterous. Okay, so we don't do that. So what do we do instead? Well, you can carefully look at family history, right? And you can kind of do this in two different directions, right? You can look at an individual and look at the generations before them, or you can look at an individual along with the generations before them to try to predict what is the risk of, whatever condition with future children. Okay, so our little key for how to read a pedigree is up here. Okay, so the circles mean female. The way that I always remember that is girls are curvier. Okay, so circles mean female, squares mean male. Okay, when you fill in the symbol, the shape, with color, that means whatever trait we're trying to track, that individual is expressing that trait. Okay, a horizontal line, right, means what does it say? Horizontal line connecting male and female represents a mating. Okay, they have babies together. In old books, it says they got married. Okay, we all know that that's not necessary for making a baby together. Okay, so when you have a line like this one that's connecting a male and a female, right, or this one, right, that means they got babies together. Okay, and then the vertical line that drops down represents the children, okay, from that mating. Now, siblings are often connected by this like a bracket like this. Okay, so grandma and grandpa had two daughters. So there's one daughter, there's the other daughter. Notice the two daughters aren't connected like this, because that would mean that they mated. And at least as of right now, given the technology that we have, it is not possible for two females to genetically produce offspring together yet. Okay, it might be in the future, but not right now. Okay, so they're not connected by a horizontal line, they're connected by a bracket to show that they are both the offspring of grandma and grandpa. Okay, all right, so that's how you read this. Okay, so like practically speaking, what are we gonna, why is my eraser not wanting to, there we go. Okay, so practically speaking, what do we want to use this for? All right, so we can use this to answer all kinds of questions, right, to like problem solve, to like sleuth. Okay, sometimes you can't get answers, but sometimes you do. Okay, so we have a condition, whatever it may be. They're using A, big A, little a. So little a is the trait. Okay, so let's just pick albinism because it starts with A. Okay, so let's say we got these two grandparents, right? And they both have a normal phenotype, right? So we know they got big A's at least. And it turns out that one of their daughters ends up having albinism. So she's homozygous recessive. Well, that information, the fact that she has this condition, the fact that she is homozygous recessive, tells us that both grandma and grandpa must have been heterozygous, because the only way that these two people can have a child that is homozygous recessive is if each one of them is at least heterozygous. Okay. Now, according to my little diagram here, they had another daughter. So they had two daughters. Here's one. She's got whatever the trait is. Okay. This other one, her circle is not filled in. So what does that mean? That means she does not have the trait. Now, do we actually know her genotype? No, we do not. We know that she has a big A. How do we know that? Because she doesn't have the phenotype, right? That recessive phenotype. Okay. So just by looking at her, we can say, oh, she has pigment. So she's got at least one big A. Okay. But we don't know what the second letter is, because you can't tell that by looking at her. Now, because this pedigree doesn't tell you anything else about her, doesn't tell you if she has any children, right? We can't figure that out from this pedigree. Okay. All right. So now let's say here's, this woman who is affected by this condition, right? And we want to know, what is the probability of, of her passing on this particular disorder to her kids, right? So we could look at her, maybe we're doing this not as a proactive thing, but as a retroactive thing, right? She and her husband have a bunch of kids. Okay. Now he does not have the recessive phenotype. Okay. So we know he's got a big A, right? One way that we would be able to figure out his second letter is by looking at their offspring, looking at their kids. So let's say they had 4 kids. And of those 4 kids, a bunch of them had a normal phenotype, unfilled symbols, right? But one of them is homozygous recessive. Okay. So that child has the trait. And that tells us, well, you always get your alleles, one from mom, one from dad. So she got one of her A's from mom. And that means that dad has to be heterozygous because she got her other little A from her dad. Okay. So that's how we know what his second letter is. Okay. All right. Here's another example. This one's a little less cluttered, right? It's just circles and squares and stuff. Okay. So we got a first generation, second generation, third generation, right? So in this case, we're looking at lobes. Okay. So remember that having free earlobes, that's with the lobey lobe, is dominant, and having an attached earlobe is recessive. So we're coloring in if somebody has the recessive condition, right, which is the attached lobes. Okay, so recessive, dominant. Okay, so here we have a family tree, right? And this is actually interesting because we're dealing with, a little bit of like kind of 2 families, right? So we have one couple, and then we have another couple. Okay, so before we know that, I'm going to just like block it out. Okay, before these people had any kids, we might not know. We might know, oh, we can see the lobe, right? We can see the free earlobe. So we know that they have one big F. We don't necessarily know what the other one is, though. Okay, but as soon as they start having kids, right? So in this couple, okay. Oh, look, here they have 2 kids. Are these sons or daughters? Okay, they have 2 sons that have attached earlobes. And that tells us both of those parents have to have a little F. So they both had to be heterozygous. They both had to be carriers, yeah, in order to have kids that have two little F's. Okay. Now, this individual, this circle over here, male or female? First of all, that person is circle, so it's female. And the shape is not filled in. So that means what kind of earlobes do they have? Free or attached? They got free lobes. Okay. Do we know their genotype? Do we know? Are they big F or big F little f? Do we know? We do not. Why not? Why can't we tell? Well, remember that I'm just going to do a quick Punnett square. Right? If you have two heterozygous parents, they can produce offspring that are heterozygous or homozygous dominant. So with this woman here, we don't know which one she is, right? And since, at least according to this pedigree, she has not had any children, right? We can't look at her children to figure that out. Okay. How about this person? Why do we know? How is it that we are able to determine that that person, who, by the way, male or female? Male. How are we able to figure out his genotype? Why do we know what his genotype is? Well, we know he has one big F because we can see he's got dangly lobes. Okay. How is it that we know that this is a little f? Well, just so happens that he and this young lady, right, are parents to these two kids, right? Okay. Now, how do we know that this person is heterozygous? Well, we know that this person is heterozygous, right? So first of all, we know there's a big F because, so let me do it this way. Okay. I'm just going to cover up what we don't know right away, and then we'll go back and figure out how we know it. Okay. So we know that this person, this woman, is heterozygous because one of her parents had attached lobes. Which parent? Mom or dad? Dad. Okay. So since dad had attached lobes, we know that all dad could give her is a little f. So we know she's got a little f. Cool. Okay. What about this woman? Are we able to tell if she's heterozygous or homozygous dominant, right? So we can tell by looking at her that she's got the, free lobes, right? We can tell that by looking at her. Do we know what her second letter is and why? Well, since one of their daughters also has attached lobes, we know that she's heterozygous because, right, kid gets one F from that parent, has to get the other little F from that parent. That's how it works, y'all. You can't just make new stuff. I mean, you can. That's a mutation, but. Okay. So that's how we know this person's genotype and this person's genotype. Okay. All right. How is it that we can figure out this person's genotype? Well, if he and she mate, right, and have a couple of kids, so boy, girl, right? The boy has attached earlobes. What does that tell us? Well, we already knew that she had a little F. But that also tells us he must be heterozygous too, because the only way this child can get two little F's is if it got a little F from one parent and a little F from the other parent. This one, we don't know. And the reason we don't know is because, once again, if we have these two parents that are both heterozygous, they can produce any combination. So we could find this out if she grows up and has kids, maybe, or maybe not. I don't know. We'll see. Okay, so that's what we use pedigrees for. Now, this is a pedigree of a standard, what we call an autosomal trait. Okay, Don't worry about that word autosome for a second. Okay. We'll come back to that. Okay. So that's how you do a pedigree. So something to practice, something for us to practice. Okay. We're going to do a practice together. It's going to be a good one too. Okay. Other things. Now we're going beyond Mendel. There is such a thing as a condition called incomplete dominance, which is essentially exactly what it sounds like. When you have incomplete dominance, what that means is that one trait is kind of dominant over the other one, but not completely. So the way that I like to think about incomplete dominance is I like to think of it as the heterozygous individuals, right? So heterozygotes have intermediate phenotypes. Ugh, can't write. I'm having problems today. Phenotypes. There we go. Okay. So here, this particular type of flower is called a snapdragon, in case you were curious. And snapdragons have this interesting incomplete dominance situation where there are two different alleles for flower color, right? So the gene is the flower color gene. The alleles are for red and for white. Okay. So if a plant has red flowers, it's homozygous dominant for red. If a plant has white flowers, it is homozygous recessive, two little r's, because remember, R, big R for the dominant trait, little r for the recessive trait. Okay. What's interesting, though, is that in snapdragons, instead of heterozygous individuals, right, here's our heterozygous individual. Instead of heterozygous individuals, right, this individual is R. Why are we doing that? RR. This one is also RR. Instead of appearing red, because it's incomplete dominance, right, they have this intermediate phenotype. So halfway between red and white is pink. Okay. All right. So that's incomplete dominance. Okay. You'll see some examples, right? What do I want to say about this? Often the way that I think about this is that the dominant allele covers up the recessive allele, kind of, but not completely, since the name incomplete, right? So the red is kind of covering up the white, but it's not completely covering it up. It's like they're mixing. It's like they're blending instead of covering up, right? We see this a lot with colors and shapes and stuff, okay? So that's incomplete dominance. Here's a human example of incomplete dominance, right? So there is a particular cholesterol disorder where people can't make enough or can't make any LDL receptors on their cells. So low density lipoprotein, that's the name. It's a type of cholesterol. Okay. If you don't make enough receptors, you have higher cholesterol levels in your blood, which can cause all kinds of nasty plaques in your arteries and clogs and all kinds of bad things. Okay. As it turns out, this particular disorder has incomplete dominance because there is heterozygous individuals have a intermediate phenotype. Okay. So most people are homozygous dominant, which means you have the ability to make those receptors on your cells. Perfect. Okay, good. If somebody is homozygous recessive, they cannot make those receptors at all. So they're going to have very severe disease because they have no way of sort of controlling this particular type of cholesterol. Okay, so they're going to be a very high risk person, right? Whereas somebody who is heterozygous, they can make receptors, but they can't make the number that they're supposed to make. So they still have the disease, right? But it's, less severe. So incomplete dominance. Okay. Interestingly enough, my this particular disease runs in my step family's family. right? So my dad's wife found herself with 2 kids, and she became a widow very unexpectedly. Her husband seemed very healthy. He was very active. He was a runner, and he was running a race. I can't remember if it was a 10K or a half marathon or what, but he was running a long distance race, and which he had done before. Like, this is normal for him to do this. And he just like had a massive heart attack in the middle of it and passed away, completely unexpected. Well, it turns out, right, that he had, even though he was in his 30s, young, right, otherwise seemingly very healthy, turns out he had ridiculously high cholesterol because he had this disease, and they didn't know. They didn't realize it because he was otherwise healthy. He was active. He was a runner, right? They didn't think, there was no reason to think that anything was wrong with him, right? And so, because it's inherited, my stepbrother has a mild version of this too. Yeah. Anyway, okay. So, so that's incomplete dominant. So dominant kind of covers up recessive, but not completely. So you end up with this intermediate, or this blending, essentially. Do not confuse all of this with codominance. So incomplete dominance, remember, means that heterozygous individuals have an intermediate phenotype. Okay. What happens in codominance is you have more than one dominant allele. That's supposed to be a no. Okay. This is the situation that we see in blood types. Okay. So we're not going to get into what the pluses and minuses mean. I mean, you can ask me in Zoom if you want to talk about it. We can totally talk about it, but I'm not going to talk about it in here because it's a different thing. Okay. So your blood type, O positive, A negative, AB positive, whatever it is. Okay. We're not going to talk about the plus minus. That means something completely different. It's unrelated. They just stick it on there because. Okay. We can talk about it later, but I'm not going to talk about it here. So all we're going to talk about are the letters. Okay. So what are the different blood types that exist in humans? So our phenotype, or our blood type, can be O, it can be A, it can be B, or it can be AB. All right. So what does this mean? Well, there's actually 3 different alleles that we see in blood type, in ABO blood type. Okay. One of those alleles is the A allele. One of those alleles is the B allele. Okay. Why are we using the letter I? It stands for immunoglobulin, which you don't care about, but it has to do with the blood. Okay. So the deal with this is that these are both equally dominant. So neither one covers up the other. Okay. But this one is recessive. And what you're looking at in this table over here is you're looking at all of the different genotypes you can have for each phenotype. So if your blood type is A, my blood type is A, okay? What that means is I could be homozygous for the dominant allele, A, or I could be heterozygous where I have the dominant A allele and then I have that recessive allele, but the A covers up the recessive one. Yeah? I don't know which one I am, by the way. I might find out once I find out the blood types of my kids. But even then, I'm looking at combinations of me and dad, and I don't know. Anyway, okay? But I might find that out, okay? Might not, okay? If you are somebody who has a B blood type, same kind of idea. You can be homozygous for the dominant B, or you can be heterozygous. Now, I don't know why they're not showing this way, because that is more common to show the dominant letter and then the recessive letter. I don't know what they're doing, but okay. Right? There's only one way of having a type O phenotype, and that is if you're recessive for both. Now, in this particular case, O, you shouldn't think of O as being like a letter. Think of O as being like none of the above, like 0, okay? So you have neither the A's, nor do you have the B's. So since it's neither, you're an O, okay? Or somebody can be AB, meaning they have one of each of the dominant alleles. Okay. So codominance means equally dominant. So that means that these two alleles are both expressed. One does not cover up the other one. However, each of these do cover up this one. Okay. So A and B are codominant, and both of them are dominant over the recessive. Okay. All right. Shall we do a blood type? Shall we do a Punnett square with this? Maybe we should. Here's an example. Okay. By the way, this is what people used to use for like paternity stuff, right? Nowadays, they have these super fancy DNA tests, which are like much more certain, who the parents are of a child. But back in the day, one of the ways that you couldn't prove that somebody that that a father that a man was a father by this, but you could certainly disprove, right? Oh, he definitely can't be the father because whatever. Okay, so this used to be used a lot for that. And by the way, you will hear all kinds of stories about like kids. This is back in the day more. Most people don't do these anymore in bio labs, just because dealing with human blood is, a biohazard and all of that. But, it was fairly common in previous generations that, oh, everybody in class is going to find out their blood type today. So you prick your finger and you use this different stuff to like find out what your blood type is. Right. And then part of your assignment is to go home and ask your parents their blood type and then do a little Punnett square, right, to see, what you got. And you'd hear stories of kids going home and asking, and it turns out that but dad's blood type doesn't make any sense. How could he can't be my father? Turns out, no, he wasn't. He just, mom was sleeping around, right? Or, mom broke up with, bio dad and then met a good man when baby was little who just decided he was going to step in that role and be dad. And they never told the kid, and drama. Okay. All right. So anyway, all right. So let's do a Punnett square. So let's say that we have a man who has type B blood and a woman who has type A. Now, this is assuming that they're both heterozygous, right? So they both have the dominant and the recessive and dominant. Okay. But we certainly could also, he could be, right? And she could be that. Okay. And so this is kind of just showing you what that would look like, right? So if each one of them was heterozygous for their condition, right, then they would have a 25% chance that they have a kid that is AB, 25% chance O, 25% chance B, 25% chance A. Okay. All right. Turns out the two most common blood types in the U.S. are O and A. So if you know your blood type, for the majority of you, it should not be surprising if your blood type is A or O, because those are quite common. B is a little less common. AB is actually quite rare. It is more common in certain ethnic groups, once again, right? So there's this kind of interesting pattern of, most of human history, we were kind of separated into, smaller communities. And so, certain ethnic groups, certain blood types are more common than others. So a lot of the Native American indigenous populations have much higher incidences of type B blood and type AB, right, than other ethnic groups. Okay. All right. Should we do like a who done it? Should we do it? Should we do it? Okay. Let's make up a story. Okay. Let's do a white screen. Okay. So let's say we got a mom and she has type O blood. I'm totally making this up as we go. Actually, I'm going to write it this way. Dad, Okay. So let's say we got a woman, right, who has type O blood. She has a baby that has type A blood, right? And she's accusing a guy of being the father of her baby, and this is before we had good genetic tests, right? So she demands that he have a blood test to find out his blood type, and it is type AB. Is it possible that this man is the father of this child? How would we figure that out? Okay, so there's our Punnett square. All right, what is her blood type is type O. So what is her genotype? That's her phenotype. What's her genotype? So remember, O is 2 recessives. He's AB, which is that whole codominant situation, right? Let's fill down. And remember, we don't have to do this one since it's the same. And then filling down and across. Filling down and across. Okay, so those are the genotypes of their offspring. What is this phenotype? What is this phenotype? So this is type A. This is type B. So is it possible that that man with the blood type of AB is the baby's father? Yes, it is possible. There are other situations too, right? You could end up with a child that has type A blood if the dad had type A. Yeah? Okay. Let's say she accuses a different guy, right? And she accuses a guy who has type B blood. Well, definitely no, right? That's also possible. If you don't believe me, do the Punnett squares. Okay. Anyway, that's okay. Practice these a little bit. Okay. Now, what's the deal with blood types? Why do we care? The reason that we care is that what your blood type is impacts who you can donate blood to and who you can receive blood from if you are in need of a blood transfusion. Okay, so what the A and B refer to is these little markers on your red blood cells. Okay, and so if you have type A blood, you have these little markers on your red blood cell that they're representing as little triangles. Okay, if you have type B blood, you have markers that they're representing as little circles. If you have AB, you have both. If you have O, you have none of them. Okay. Now, here's the thing. One of the unique characteristics of our immune system is that our immune system makes these proteins called antibodies. And what antibodies do is they attack things in your body that don't belong there. Okay. So if you are somebody who has type A blood, then your immune system knows that you have this little marker, the triangle shaped marker, but it would see these round markers as foreign and thereby attack them. Okay. So the antibodies that are present in the blood of somebody who has type A blood are anti B. So I have type A blood. So what that means is that I have antibodies in my blood that if I was exposed, if I got a blood transfusion from somebody who had the round markers on their blood cells, my antibodies would attack those. Okay. And it causes clumping and bad things. Okay. The opposite is true for people who have type B blood. They are anti A and anti, or because they have the B markers, their body represents that. I can't talk. Their body recognizes that as this belongs to us, but the triangle shaped ones, the A, they recognize as foreigns. They have anti A antibodies. But what if you're AB? Well, aren't you lucky? right? Because you have both markers, you don't have antibodies that will attack either because they're both self, right? So you don't have any antibodies, okay? What if you're O? Well, if you're O, remember, you don't have either of those markers. So you have antibodies for both, against both. Okay. So when we talk about blood types, one of the things that we often say is we describe one of these blood types as the universal donor, and one of these blood types is the universal recipient. Okay. So let's say that you get in a horrible car accident and you need a blood transfusion. And in the moment, right, they need to get blood into you as soon as possible. They don't have time to like do a blood test and like check your blood type, right? We need to get blood into you immediately. So what are they going to do? Well, they want to give you something that your body is not going to react negatively to. Okay. So what they do is they give you blood that doesn't have any of those little markers, right? This is why in all the doctor shows and the hospital shows, right? ER kinds of shows, right? If you don't know somebody's blood type, you give the type of blood that you give in an emergency that you give to that person is O, because O, nobody's going to react to O, right? The O blood type offends no one. Okay. So O negative, right, is the universal donor, meaning that anybody can receive O negative blood and not have a negative reaction. Okay. Who is the universal recipient, meaning they can receive any kind of blood. It's not going to cause problems for them. Who's the universal recipient? AB, right? Because they have both the triangle markers and the circle markers. They don't have any antibodies against any of those things, right? So they can get any type of blood and they're good to go. Okay. So one interesting thing to think about is if you know your blood type is to think, okay, what kinds of blood could I receive if I needed to, right? If I had to have a blood transfusion. Okay. So since my blood type is type A, what that means is that I can, of course, get type A blood, right? But I can also get type O because that's not going to offend my immune system. Okay. If somebody is type B, what kind of blood can they get? They can get type B or they can get type O because that's not going to offend anything. What if you have type O blood? So my husband has type O blood. What kind of blood can he receive? He can only get O, right? Because his antibodies are anti-A and anti-B, right? So his body would have a really negative reaction if he got either A blood type, B blood type, or AB. That would cause problems for him. Yeah. And so if you donate blood, and I highly recommend that you do if you're able, because we always have need for blood. They'll bug everybody, right? Once you do it once, then they keep like calling you up. They're like, come on, please come back, give us more. Because so few people donate that there's a lot of blood shortages. But certain groups will get bugged more than others. So if you happen to have type O negative, right, they're going to be bugging you all the time, right? Because we have a greater need for O negative blood because in an emergency, that's what you give to people, right? When you don't have the time or you don't want to trust making a mistake and making something worse, right? So you just do O negative in an emergency. Okay. So people who have O negative blood get called a lot, or if people have rare blood types. So B is rare, AB is rare. And so those people are more likely to be called just because there's not as many people that have that. So in planned surgeries or planned transfusions, right, then they want to match your blood type because why give you O if, they can give you one that matches you. And if you have a particularly rare blood type, right, it's nice if we have a stockpile of some of those. Okay. Anyway, so that's blood type. So codominance, blood typing is codominant. All right, last two more things in this video, and then what are we gonna do? Yeah, we'll stop it off. We'll stop it. I can't talk. We're gonna talk about two more concepts here, and then we'll have one last video. Lots of stuff in genetics, you guys. Okay. Pleiotropy is a situation where a single gene has multiple effects on the body. right? So most of the things we've been talking about so far, right? It's like, oh, this gene affects your earlobes and just your earlobes, right? This gene affects your, coat color and just your coat color. And I'm going to pause because somebody needs. Okay, wrap this up because I got to go take my kids somewhere. Okay, sorry. So pleiotropy is when one gene has multiple effects. So most of the things we've been talking about up until this point, that's, each gene has only one effect. An example of pleiotropy, and there's others as well, but one example of pleiotropy is the gene that causes sickle cell disease. Sickle cell disease is a condition where people who are either homozygous for sickle cell or even heterozygous have some proportion of their red blood cells, instead of being round, have a sickle shape like that. Okay. Well, that has all kinds of effects on the body. It makes you anemic. It makes you weak. It causes pain. It causes organ damage, right? So the one gene has multiple effects. Okay. Pleiotropy. Not to be confused with polygenic inheritance. Okay. So the thing to know about polygenic inheritance is that polygenic, you know what poly means? Poly means many. Genic genes. Okay. So in polygenic inheritance, this, by the way, is the way that most characteristics are inherited. It's actually multiple genes that together are responsible for one trait or character. Okay. So adult height is a perfect one. Okay. So there's actually lots of different genes that play a role in this. Yeah. And so there's a big spectrum of heights. It's not like you are either tall or you are short. Those are the two options, right? Whereas in all these other examples, it was like you're either this or you're that, right? You either have attached earlobes or you have free earlobes. Okay. In polygenic inheritance, it's actually a bunch of different genes that are responsible for a particular characteristic. These are harder to figure out, so we don't do problems about these, right? So height is one example. Another example is skin color, right? So there is once again, a wide range of skin tones. It's not like you are either black or you are white, right? Like, and that's it. There's a huge range of all kinds of different shades of beiges and browns, right? And the reason there's a range is because there's actually multiple different genes that are responsible for that, right? And so there's a range in color. And quite frankly, one of the reasons it bothers me that they use the labs as your example, the dogs as your example in there, is that somebody, one of you, during that discussion of the labs is like, but I have a yellow lab. What's the deal with yellow labs? Why aren't there yellow labs in here? Well, as it turns out, coat color in most animals is not just caused by one gene, it's actually caused by multiple genes. It's actually a polygenic trait. Okay. So how do you get a yellow lab? Well, now this is going to blow your mind. Okay. A yellow lab is simply a black lab or a chocolate lab that has a different gene which suppresses their coat color. And so they end up with blonde. So it's a whole separate gene. Yes, I hear you. I'll be there in a second. Okay. So that's polygenic inheritance. Okay. This is where we're going to stop this video. The next one is where we'll finish. Okay. So one more after this.